The information covered in these notes is Chapter 11.4, which talks about a cellular division process called meiosis. Meiosis is different than mitosis. In mitosis, we started with a cell, and let's say the cell had the human amount of chromosomes, which is 46. At the end of cell division, you're going to have two cells that are identical to the parent. And so the two cells also have 46 chromosomes. So in mitosis, the division is asexual because you've got one parent cell, and the daughter cells are identical to the parent. Now, in meiosis, we have something a little bit different. And I'm going to just give you a summary here. We're going to start off with 46 chromosomes. We do have one parent cell. However, through the process of two cellular divisions, we're going to end up with 4 daughter cells. 1, 2, 3, 4. And each of those daughter cells is going to have half the amount of DNA as the parent. This is called meiosis. And none of these cells is identical to the parent. Each of these daughter cells is unique compared to each other and to the parent cell. So where mitosis is considered asexual reproduction, because you're getting daughter cells that are clones of the parent, meiosis is sexual reproduction or involved in sexual reproduction because you are going to get daughter cells that are unique compared to the parent. And ultimately, two parents will donate their DNA to have the offspring. So I'm jumping ahead, but I wanted to kind of preview the differences between mitosis, which we've already learned, and meiosis, which is going to be new for us today. So to backtrack a little bit, almost every organism is a combination of both of their parents. Half of the chromosomes are going to come from the mother, and half of the chromosomes are going to come from the father. Now, with humans, and I'm going to talk mostly with regards to humans, we have 46 total chromosomes in each cell, except for a couple of exceptions, but I'll talk about those later. So if we take a sample of your tissue, most of your tissue is going to have 46 chromosomes in each cell. And of those 46, 23 came from each parent. So your father donates 23 chromosomes, and your mother donates 23 chromosomes, and then you have 46. When you have a chromosome and a corresponding chromosome from each parent, that set is called a homologous pair. And just for consistency, I'm just going to go back to blue. Homologous pair. So the chromosome that comes from your mom has a set of genes on it, and it has a counterpart chromosome from dad. And that is shown here. So you have, let's say, this chromosome, and it's bent, but that doesn't mean anything. I'm going to shade it in blue, and we're going to say that that chromosome is chromosome number 1, And the blue is just going to represent that dad gave it to you. And chromosome number 1 has a counterpart that mom gives you. And so together, this is a homologous pair. The same is for every pair of chromosomes through the sex chromosomes, which are at the end. So you have 22 pairs of chromosomes plus a pair of chromosomes that determine your genetic sex chromosomes. And 1 through 22 are called autosomes. So these chromosomes do not influence anything about male or femaleness. These are going to code for traits like amylase and the fact that you have keratin that makes up your skin and your hair, melanin, that's pigment in your skin, catalase. So genes that code for you being a human being, irregardless of your genetic sex, are coded for on the autosomes. The last pair are called the sex chromosomes because they determine your genetic sex, male versus female. Males have an X and a Y chromosome, and females have 2 X chromosomes and no Y. We'll spend more time with the sex chromosomes and information coded on the sex chromosomes another day. The reason why we're talking about this picture of chromosomes, which by the way is called a karyotype, is because it helps to understand homologous pairs and the number of 46, because meiosis is going to take this number of 46 chromosomes and reduce it. So meiosis is a process by which the cell divides and reduces the chromosome number from 46 to 23. So moving down, remember, with each pair of chromosomes, so I'm just going to draw a circle around a pair. 1 of these chromosomes comes from mom and 1 comes from dad. And the verbiage for that is a homologous pair. If a cell has homologous pairs, so it has 46 chromosomes, the adjective to describe that is called diploid. Diploid is an adjective. It is not a noun. So in order to use it in a sentence, you would say the diploid cell. It describes the cell. So when I see the word diploid, I know the cell has 46 chromosomes. It has homologous pairs. Again, the diploid number in humans is 46. And almost all cells in your body are diploid. Skin cells, muscle cells, your organs like your liver, neurons, cells that line your respiratory tract, cells that line your digestive tract. I mean, most of the tissue that makes up your body is diploid. So if we were to look at the nuclei in one cell, and we could actually put a chemical to condense the chromosomes, there would be 46 chromosomes in the nucleus. In your respiratory tract, in the nucleus, there would be 46 chromosomes. Now, let's talk about cells that are not diploid. There are some cells in your body that do not have the full set of chromosomes. They only have half, 23. Cells that only have one set of chromosomes are called haploid instead of diploid. And I remember that because kind of sounds like half. So haploid, again, is an adjective. So in a sentence, you would have to say haploid cell. And that adjective describes the cell. And by saying that word, I know that it has only 23 chromosomes and it has no homologous pairs. These cells in your body that are haploid are sex cells. And the proper term for a sex cell is called a gamete, Gamete. The examples of gametes in most sexually reproducing organisms are called eggs and sperm. Now, if you were a plant, we call eggs ovules and we call sperm pollen. So fun fact, pollen in plants is plant sperm, and pollen is haploid. The ovules in a flower are haploid, and so when pollen pollinates a flower, the pollen is going to fuse with an ovule, and you are going to get a seed. So eggs are typically made by the female, and sperm is made by the male. And in order to have the next generation, the sperm is going to fertilize the egg, and the 23 chromosomes in the sperm are going to fuse, or I don't want to say the chromosomes are fusing, but the nucleus is fusing with the nucleus of the egg that has 23. And you end up getting, oops, and I put that 23 in the wrong place, sorry. You end up getting a zygote, which is the fertilized egg when the sperm and egg fuse, and that restores the number of 46. So I kind of answered my own question here, but essentially I was going to say, why is meiosis important? And the reason is if you did not reduce the chromosome number to 23, if let's say a sperm had 46 chromosomes and an egg had 46 chromosomes, upon fertilization, all of a sudden you'd have 92 chromosomes, and that would be bad. Organisms typically do not like to have manipulations done to their chromosome number, especially humans. Oftentimes it is fatal. So by doing this reduction division, you ensure that the next generation has the proper chromosome number. Now the pictures here just kind of show you again that haploid, you only have one copy of each chromosome. And if you have a diploid cell, you have two of each kind of chromosome. The haploid number here would be 1, 2, 3. There are 3 chromosomes. The diploid number would be 1, 2, 3, 4, 5, 6. There are 6 total chromosomes, but 3 pairs. So, of course, if you have 2 of each pair, and there's 3 pairs, you have a total of 6. So we often will depict haploid as N and diploid as 2N. And the N means how many different chromosomes do you have? How many different chromosomes are there? So here we had 1, 2, 3 different chromosomes. We also have 1, 2, 3 different chromosomes. We just have 2 of each. So the diploid number is 2 times N, 2 times 3, which is 6. Now let's talk a little bit more about meiosis. In meiosis, haploid cells are produced in the body from diploid cells. And those diploid donor cells are going to be found in the ovary or the testes. So we are going to start with a cell that does have 46 chromosomes. And again, the process by which that cell divides and reduces the number of chromosomes to half is called meiosis. The definition given by your book is that meiosis is a process of cell division in which the daughter cells are not identical to the parent cell. Instead, they have half the amount of DNA and they are genetically unique. You can think of every egg or every sperm cell as a snowflake. Each one is genetically different from the rest, and there will never be two that are made that are identical. So in summary, meiosis makes reproductive cells only. Meiosis makes haploid cells. And meiosis makes unique cells. Meiosis is different from mitosis because it has 2 rounds of dividing. The 2 divisions reduce the chromosome number from 46 to 23. The first stage of dividing is called meiosis 1. That's easy to remember because meiosis 1 comes first. And the main thing to remember here is that in meiosis 1, the homologous pairs are separated. You should probably take your pen and put a star next to this statement because this is the most important thing to remember about what is happening in meiosis 1. The homologous pairs are being separated. Now the picture below shows an overview of what's happening in meiosis 1. And really what's going on is the same phases you learned in mitosis are still occurring, and they are very similar. However, there's a big difference. The chromosomes find their homologous pair in prophase, and that never happened in mitosis. Other cellular changes are similar. You have in prophase, the spindle forms, the nuclear membrane goes away, the chromosomes condense, but the fact that the homologous pairs find each other is new, and that's never happened before. When the homologous pairs find each other, we call it a tetrad. And the chromosome from dad, it's a replicated chromosome because before meiosis, DNA still is replicated in interphase. That is the same as mitosis. You still had replication. So we're still going to have replicated chromosomes. But the chromosome that comes from mom and the chromosome that comes from dad find each other and they pair up. And we call it a tetrad. And what ends up happening is little pieces of mom's chromosome and dad's chromosome switch places. Now you might be wondering, why the heck does that happen? This is what makes you unique. The fact that pieces of mom's chromosome switch place with dad's chromosome ensures that the resulting egg or sperm is going to be unique. And we call this crossing over. Crossing over takes place in prophase 1. Now, the number of sections that switch places between homologous pairs is random, and where the crossing over takes place is random. But if you can imagine, 23 pairs are going to be engaged in crossing over, so you are going to get a lot of switching of genetic information between mom and dad's chromosome. Then the pairs line up. In mitosis, the pairs did not line up. All 46 chromosomes lined up individually down the middle of the cell, and there was no crossing over. But in meiosis, the pairs found each other, crossing over occurred, and then the pairs line up down the middle of the cell. And here is where we get our very important star. The homologous pairs are separated or pulled apart. So at the very end of meiosis 1, you end up with 2 daughter cells and they, believe it or not, are haploid already. Because by definition, haploid, even though I told you it was 23 chromosomes, it also means that there's no pairs. And since we separate the pairs in meiosis 1, technically these 2 cells are haploid. But we still need to pull apart the sister chromatids. And that is what meiosis 2 is all about. In meiosis 2, the sister chromatids are separated. Now, we call this meiosis 2 because it happens second after meiosis 1, and we still have the same 4 phases: prophase, metaphase, anaphase, and telophase, just like we had those 4 phases before. But we distinguish them by saying prophase 1, metaphase 1, anaphase 1, telophase 1, and prophase 2, metaphase 2, anaphase 2, telophase 2. Meiosis 2 is going to look a lot more like mitosis, but remember we no longer have pairs. So the resulting 4 cells at the very end are going to be unique, and they are still going to be haploid daughter cells. So at the very beginning, we started off with 4 chromosomes in this particular picture. And how you count, I'm going to zoom in, is the trick is to count centromeres. 1, 2, 3, 4. The number of this cell, the diploid number, is 4. The daughter cells at the very end of this meiosis diagram, the haploid number is 2, and we're going to count centromeres. 1, 2. So we have 2, 2, 2, 2. So if you start off with 4 chromosomes at the beginning of meiosis, you're going to end up with 2 chromosomes at the end. In a human, you'd start off with 46 chromosomes, and the resulting gametes would have 23 in each. I would advise you to read what happens in each of the phases just to get used to what's occurring and be able to visually recognize what phase each of the cells is in. But a big clue is if you see homologous pairs being separated, it's meiosis 1. And if you see sister chromatids being separated, it's meiosis 2. And that is it.